Thank you to CEWiT first off for inviting us to be here and for providing this forum for all of you to have an opportunity to hear about what's going on campus and ways we can engage. And thank you everybody for coming and being on the panel, and for all of you for attending with us. So let me start by just doing some quick introductions of everybody, and we'll start at the end with Keith Davis. He's the director of the Johnson Center for Innovation and Translational Research. Next is Roger Ennis. Roger is a professor in the Department of Biology. Followed by that is Andrea Hohmann. She is the Linda and Jack Gill Chair of Neurosciences and Professor, Department of Psychological and Brain Sciences. And then we have Hannah Block. Hannah is an assistant professor in the Department of Kinesiology, and Yvonne Lay, research scientist with the Department of Psychological and Brain Sciences. I'm going to ask that we start today by having each of you provide a brief introduction of yourself, a little bit about how you categorize yourself in your research, and whether you're new or experienced in faculty and industry collaborations. And I'll start with you, Yvonne.
Okay, I'm a neuroscientist by training. Actually, right after my postdoctoral work, I went first of all to a CRO, which is a contract research organization that develop assays. And then I went to ICOS for about 10 years. ICOS is a company that developed Cialis. I wasn't on the Cialis team. I was on another project. But while I was at ICOS, I both worked with CROs as well as academic collaborators, where we have many years collaboration with academic labs, where we basically take some of their interesting ideas and then evolve, and then we work with them to file patents. So it's kind of very much a two-way. I came to Bloomington 2007, partly for family reason. My husband and I were both recruited here. So I was now back in academics and learning how to write grants, and started working with Andrea. So because partly or mainly because of Andrea, I think I am now very good at grant writing, both working with Andrea. We have so far got every grant funded, if not always the first time.
No,
but
some version of the grant.
Some version. That's
right.
Don't say that too loud.
Find some wood, that's not that.
We're very persistent. But also, again, with starting collaboration with Annetta Shaker at Psychiatry Department in IU School of Medicine, and also from some of the work with Andrea, Annetta actually started the company called Antigen first, and then he approached me and then Andrea to have some of the project that actually originally started at ICOS but continue in Andrea's lab. So we used that to start the main focus of that for Antigen. So we wrote a 2-year SBIR phase 1 grant. We got it funded $700,000, and Andrea was with me at midnight changing commerce right before the symposium Golden Conference that she actually organized. But anyway, we managed to stay awake and have a great conference that Andrea hosted that year. So after that, we have now actually we have like 3 SBIR phase 1. So raised probably 1.5 million. So we started the company. Now I have a 60% role in that company in the incubator space in Indianapolis. We have CEO, we have CFO, they part-time, and we have about 3 full-time, plus a lot of different consultants that I recruited back from ICOS. They raised about $1 million, about a little bit more, and so we have the management team to continue to raise. So now I'm as part of the company, I also contact with CROs as well, have contracts with Andrea and then with IU School of Medicine to do different pieces of the work. So I kind of cross both ends. I'm sorry, it's a long, complicated introduction.
That's okay, thank you.
So my experience has been very different from that. So I'm also a neuroscientist by training, basic science. So I study how the brain controls movement and how sensory information plays into that. I came here to IU in 2013 for my first faculty position. And this project is by far the most applied thing I've ever done. So it's a little different for me, but it has been really good. My experience with industry partners is 0. So yeah, I guess I'm at a different end of the spectrum on that.
Well, everyone, my name is Andrea Hohmann. I was actually trained as a neuroscientist, as a basic neuroscientist. So all my laboratory work has really been focused in rodents, mice, and rats, with the primary objective of identifying novel therapeutics for pain. And actually identifying novel therapeutics that would suppress pain but lack the unwanted side effects of existing therapeutic interventions. So to do that, that basically had me not only work on pain, but also work on optimizing side effect profiles and looking at things that were really debilitating for a therapeutic intervention. The major one would be something that's typical of like opioids, like dependence. So trying to get compounds that were in novel therapeutics that suppressed pain but didn't make you high, that didn't produce physical dependence or withdrawal, that didn't produce unwanted memory and motor side effects that were typical of existing medications. So that had me, although I'm essentially a pain researcher, that had me also working in other domains that would look at drug abuse profiles, that would look at memory profiles, would look at motor side effects, and working at very different levels of analysis. So I had trained at Indiana University with Yvonne, I might say, in evolving these small molecules for this exciting target for further development. And I would say that my interactions and collaborations with Yvonne really were the first start of my official, interactions kind of with industry in that sense. But I came and moved to Indiana University in 2010 when I was recruited here as a Linda and Jack Gill chair, and most of my work actually has really been focused on cannabinoid receptors, which are the receptors which bind marijuana, and it's been focused on identifying ways to exploit the endocannabinoid system to suppress pain without unwanted side effects. So I've worked historically with a lot of different chemists. And I would say that really it was working on a very exciting therapeutic target that really had very little precedent in the literature. And I think that it's been a really interesting and fun road of discovery along the way.
So I'm Roger Innes. I'm from the biology department. And for something completely different, I work on plants. I don't work on medical applications. And I've been at Indiana University for 25 years. And pretty much that whole time I've worked on very basic research, trying to identify genes in plants that mediate defense responses to infection by bacteria, fungi, and viruses, with the goal of really trying to take advantage of existing plant genetics to develop crop plants that are naturally resistant to disease so that we can reduce our dependency on fungicides and other pesticides. And so for 20 years of that research, it was extremely basic, where we're just identifying genes, looking at the proteins those genes encode, and trying to understand how those proteins work. And really was hoping somewhere down the road there might be some application of this research, but was not working directly with the company or even really trying to pursue developing applications. But about 4 years ago, my NIH funding dried up, and that made me really look at other sources of funding. And I started talking with contacts that I had in the plant biotech industry, and was able to reconnect with a former postdoc that had worked in my lab that had gone on to a research position at a large biotech company. And he said his company was really interested in some of our things that we had discovered. And so he and I put together a proposal to his company to develop a project to develop a genetic-based disease resistance to a really important disease of soybeans, which is an issue in Indiana, but it's particularly, this disease is a huge issue in South America. It's called Asian soybean rust. It's a fungus that started in Asia, jumped the ocean, infested the soybean farms in Brazil and Argentina, and they now are spending over $1 billion a year on fungicides in South America to combat this disease. So there's a huge market for it, and so that's why this company is interested in developing a genetic-based resistance, which then could eliminate use of those fungicides. And so this project then, the company was interested enough to fund development of the ideas that we have, and what's I'm really enjoying about the project is it's a collaborative research project. So it's not just the company's giving us money to develop the project in our lab. We are working with the scientists at the company's BASF, and so we're working with those scientists to help develop this project. And it's my first time doing it, but I have to say it's been pretty fun to feel like my 20 years of research might actually have an impact in the real world at some point.
So I've had a interesting career. I started out in plants and actually worked in the same field Roger did for a number of years. We were, I think, one of the first few postdocs who came out on the job market who had disease projects in model plant systems. So I started out at Ohio State and was there and ended up being director of a plant biotech program. Then I decided I wanted to try something else and joined a company, a biotech company in North Carolina, working on, again, plant biotech. And that was a very interesting process. We went through startup phase to IPO to being acquired. So from there I experienced basically everything you can do in a company, a successful company. Then after that I went back to academics and switched from plants and started working in cancer biology. So my current research, when I have time from my center activities, which is my real job, my day job, I work on peptides, natural peptides that have anti-cancer activity. And really, being on the panel here today, I'm going to talk more about that part of my life versus being center director.
So you've all kind of mentioned, except for Hannah, except you from this one, that you're working with industry. Do you have maybe a little story about how you came to identify your partner and begin that work, and kind of how that work started? Was it a difficult process to find a partner? I was kind of interested in hearing how you went about identifying your partners.
Well, I'll start on that. Because I like to tell this story because I think anything that tells you anything can happen and not to leave any stone unturned. So when I first started this cancer project, I was a plant researcher. Nobody was going to give me a grant to do this, especially NIH. So I had some funds available, so I just started doing some work. And the strangest thing happened. So I was going to isolate this peptide from soybean protein. And in the town I was at, Owensboro, Kentucky, there was a big soybean processor there. So I went to talk to their VP of production and say, well, can I get this material? They call it white flake. It's basically the crude material left over after they get the oil out of the soybean. And we started talking about it and what I was going to do. And so he was really interested, so he gave me some white flake, and we looked at it, and we could get the peptide out that we wanted. So I told him about it, and he said, "Well, this is really interesting. It's a privately family-owned company." He says, "Well, I'm really interested in cancer." Of course, like most families, he had family members who had cancer. And he says, "I want to fund your research." So I'm like, "Oh, no problem." And he says, I think we can get some money from the Kentucky Soybean Board because they fund research. So he took me to the soybean board and I gave a little presentation. I mean, normally they fund field studies, variety development, things like that. But they said, well, you know, if we can find something that fights cancer in soybean, that'll, you know, increase people eating soybeans. So they funded it. So over, you know, the past 6 years, between a couple years of funding from that soybean board and then from the company, Owensboro Grain, they've invested almost $1 million in developing this project. So, you know, I think, of course, as an academic, and particularly if you're trying to get tenure and stuff, you have to get your grants from NIH, NSF, whatever the agency is for your field. But there's a lot of sources of funding out there that you might not imagine. And I would be very open-minded and creative about where you seek funding from, because there's a lot of money out there from non-traditional sources.
Going down the line?
Sure.
So I already mentioned a little bit about the collaboration I currently have started with a former postdoc of mine who joined that company. But I should say that was not the first company that I sought funding from. And the kind of lesson I learned, I think, was that you really need to make those personal connections when you're looking for funding from non-traditional sources. So through activities at Indiana University, we had made contacts with scientists at Dow AgroSciences in Indianapolis. I had some contacts at Monsanto, and I sort of shopped my idea. We had a preliminary patent application already on our system, and frankly didn't get anywhere with those folks. And it was only when I had somebody inside the company that was really excited about the project that they were able to sell it to their management and put a project together. But even at that phase, and I think this is different from what Keith just talked about, because we're talking about large companies here. And for large companies, their process of deciding what academic labs to fund is not quite as nimble as some family-owned company.
Right.
But anyway, from the time that this former postdoc and I decided we were going to try to get his company to invest in this project, it was over a year before we had a contract in place. And that was mostly due to lengthy negotiations between their lawyers and IU lawyers over the terms of the contract. And about the only lesson I could take from that is it takes patience. And I owe a lot of thanks to Keith to keep that whole process going forward. And, making phone calls with representatives from the company to say, hey, what's going on here? We haven't heard from you in a month. Can we move this forward? So certainly it was really helpful to have the Johnson Center and Keith kind of behind me to make the process happen. But once everything was in place, it's gone really smoothly. So, it's, like I said, I'm enjoying that collaboration.
Well, perhaps I'll just share a story of probably like Keith, maybe my initial foray into collaborating on a project that was submitted through an SBIR grant had to do with wanting to avoid a spurious study section at NIH that was, in the paint field, for example. And so I think it's kind of funny when I think of the history, like you've already heard that, our grants, whether, that we've been successful in getting funded, maybe not initially on the first round, that there is, I feel like that there is this element that you can be right too soon, and that you can propose an idea and you're working on like something that's like a novel mechanism, and our initial version of submitting a R21 exploratory grant for NIH for pain, that was had a good score, but skepticism in terms of the mechanism and establishing whether this mechanism, was happening in the animals, and it was sat in like limbo for about a year and was a fundable score, but was like not really assigned to the right institute in some ways, and then was pulled from this limbo state and then funded. And based on those types of experiences, it says, oh, okay, well, this is the response this grant has gotten, or this mechanism has gotten in pain. how about PTSD? And Yvonne, has started this company with an Shakar. So basically taking the same target and evolving it for a different indication, which was very novel for PTSD. And even, I can take other examples of where you can see, a subsequent grant that then I initially submitted through an SBIR phase 1 mechanism, thinking that, perhaps this grant would be a better studies section because they would people who would understand how it's not just about suppressing pain, but also reducing the abuse liability, and that thinking that, get it into a mechanism or have it evaluated in a mechanism that, has a pay line of much higher than at NIH. But then finding also that, some of the valuable feedback from that type of a review was then incorporated, and then the grant that I actually got funded was then funded through an NIH Cutting Edge Basic Research Award, which was a non-SBIR grant mechanism, but went through various evolutions to kind of find the right study section and find the right fit.
You, John, to jump to?
Yeah, I think we'll jump in.
Okay. I should comment that when I was in ICOS, I also, as a scientist, evaluate projects that were proposed to ICOS from other companies. So whether it's academics or in that case, companies. And because you usually only see 1 or 2 page thing, and it always seems like some company or some academic is something they're going to cure, generate world peace or whatever. So you tend to be more skeptical. And certainly there were evaluations where we got to a point where we have like all the senior managements, we have this whole table of all the senior managements, and we finally get to a point where we look at all the data, because in the beginning you have the business people have all this like Salon and Downs thing, and then you look at it and you find out some of the key experiment, only 1 out of 3 works. And you say, why are we wasting our time? So certainly from what Roger said, that if you have, it's always about personal, connections. So if I were in this stage, and some of the people that I did my postdoctoral with, or whatever, people that I actually have very high respect come with me with an idea, I'm more likely to say, okay, I think we need to really look at it because, I know the quality of that person's work, versus someone they come in that may even have a more promising stuff, and you kind of like, well, yeah, am I going to waste my time? So certainly that part's important. But setting back now to with IU, since I kind of am both ends, right? I'm half this company, half IU. Because of some of the recent difficulties of some of the fairly major PI in negotiating with IURTC in terms of, licensings and what IU owns and not, IU is getting a reputation of being like, if you be in a bench and do anything, and even you take a breath or you use IU database, IU owns everything, then certainly from the company perspective, when I guess eventually we talk about is intellectual property, you want to protect. So I'm now in both ends, but speaking from a CEO of the company, they want the company to own all the rights, partly because once you get into multiple university, it just gets difficult. And even in this case, the company Energen is owned, I think by now still like 15-16% by IURTC. So they own the company and yet they make it difficult, so it gets very kind of complicated negotiation. So the simplest deal, and we're hoping and we're going to be working additional contracts of IU to work with Keith, where we lay it out where, okay, the company wants certain rights. What does the PI want? Do they want a fair contract to pay for all the expenses? Do they want to be a paid consultant? Do they want to have business development is like anything works. So a lot of times you get milestones, so that you don't. And I've talked to, and I have a lot of friends that are like me who have gone to industry, and a lot of them left research to be in tech transfer. So I actually have quite a lot of friends that had transfer of some of the major institutes here. And the more savvy the company, the universities are, where they are like, you know, in medical school where it seems like every PI have a patent and start a company. Their goal at tech transfer is to have the minimum bar. So have it all, like, because a lot of stuff won't work, right? You know,
Right.
so have it so-called back-ended. So if your stuff work, if it get to the clinic, then you get huge payoff of percent, right? You know, whatever. I mean, it's all how you want. So the PIs have to think about, what do you want? Like does Roger think, well, I want my, you know, invention to be out in the world. You know, I do want to own something. I don't want to give it away, but, you know. But if you spend a lot of time arguing about, like, how much, so I would tell you at ICOS that a lot of universities will just won't touch because the tech transfer is so difficult. Even if the PI said, you know, I'm fine with this, but so I just want to say it's as much an environment that, of course, Keith is, you know, key to, and I should say, Joanne, the center is try to break those barriers and have the IU and the PI say, come on, you know, let's find what you really want and make everyone happy. And I think that's the tricky part, and so that's my comment from being in different side of the coin.
I want to come back to that in a second. Before I do, Hannah, can I ask you to talk a little bit about your project? Because I think everybody else has kind of talked about theirs within the other questions, and we haven't had a
Right.
chance to hear about what you work on.
Right, that is true.
And what you work on is interesting.
So what I'm working on grew out of my basic science research in humans, where for various reasons we do careful measurements of movement and sensory measurements, especially position sense, which is proprioception. So we care a lot about measuring it accurately because it's not the cleanest signal, it's very noisy. If you want a useful measurement, you have to really, really be careful and get lots of good data. I learned eventually, although I don't have a clinical background, a lot of my stuff you know, could eventually maybe influence how we rehabilitate people after a stroke, for example. And I got more familiar with the clinical side of things and learned that when clinicians measure proprioception, which is very important in a number of clinical populations, they don't have a good way of doing it. So they may get a very rough measurement by having the patient close their eyes, and the clinician takes their finger and moves it up or down and says, did I move your finger up or down? And of course that's not standardized at all. And they're going to move different amplitudes and different speeds, both of which affect proprioception. And they're going to do it like 6 times. And if you get all 6 correct, then they'll be like, okay, you're fine. And if you miss one, then we don't know. Maybe it's not fine. It's very coarse information. So I wanted to turn a technique that we use in the lab into something that could be done in the clinic by making a tablet app with a customized stand to hold the hand in place. And I mainly focus on the hand, just because that's the thing I study is upper limb movement. And that's something that's very impaired after a stroke a lot of times. So patients have trouble manipulating objects and doing tasks of daily living and whatnot. And a complication is something like 50% of people have impaired proprioception after a stroke, but many of them also have impaired movement, or probably most of them have impaired movement. But when you watch someone making movements that are impaired in some way, you can't tell whether it's because the motor system is damaged or does the motor system have access to bad proprioceptive information? That's all you see is the output, the actual movement that's impaired. So it could be very important for determining, treatment plans and determining exactly what the functional deficits are to have an accurate clinical measure of proprioception. So with this project, we did already do just a study in controls that didn't have an actual tablet app. I bought a portable notebook monitor that looks sort of like a tablet but plugs into a computer, so I could run my own code. And we tested a bunch of control subjects and we published that data. But to move to the next step, we really needed to get clinical data. And I mean, the whole point of making it be done on a tablet is that it's portable. So we used the project to get the app actually written and buy actual tablets and a 3D printer to develop the custom stand. Because of course, how you position the hand is very important. And we wanted to make the test as standardized as possible. So once the hand is in position and you put the tablet actually goes on top of the hand, which is sort of counterintuitive, but that's how it works. Once that's done, then the tester doesn't really do anything. So the hand stays still, the patient looks at the screen and responds to different stimuli that come up automatically. And then at the end, there's a readout, numerical value of proprioceptive sensitivity and bias, which are two things you could not measure from the up or down test, and certainly that you could not quantify from the up or down test. So that's basically the project.
Great. Thank you.
I'm going to interject here.
Oh, okay, well, go ahead. You can, you're the director.
Since I'm out of slapping range.
You're not out of...
But I mean, that's what I found. This is a good example of one of the most important things I think the center does is when faculty have these more basic ideas that they've developed through the years of their basic research. If they need to do something, a prototype or do something that might make it more interesting to industry, to fund further work, we have a grants program to do that. So that we've funded this project so that, to try to get some of these prototypes done. And I think that's a, Roger had, I didn't know you were that old. Well, 20 years of research behind him that already was really clear that this was an important thing that could be done. In other cases, or depending on the industry, they may want to see a certain type of data before they'll even look at it. So we can help you generate that type of data or prototype.
Right. Yeah, I wanted to come back to what Yvonne kind of alluded to as some of the challenges and differences in this sort of work that you're doing as compared to some of the more traditional NIH, NSF, Department of Defense, et cetera, the more traditional research work as compared to your translational research work. What sort of other challenges and differences have you found? I'll leave it up to any of you who want to respond to it. Or
I guess I can start. The first challenge one I already alluded to, which was just dealing with all the lawyers to get the contracts signed. And that there's not much more to say about it other than you had to be patient and also had to keep after them. That's Keith's main job on that project, was keeping after the lawyers.
yeah, that's a service we provide, is to bug lawyers.
Yes.
A lot. We kind of enjoy it.
Service. One thing I wouldn't quite call a challenge, but it's been kind of interesting for me to see is just how different project management is in an industry setting versus an academic setting. So part of this contract that we developed, we wrote down milestones about what was supposed to happen after 6 months, what's supposed to happen after a year, what's supposed to happen after 18 months. And they actually pay attention to those milestones. Unlike when we write a grant application, we say we're going to do this, we're going to do this, we're going to do this. We might even have an approximate timeline. And at least my experience as an academic, as soon as you have the money, you do whatever you want. And you hope that you've done something interesting by the time that you need to renew the grant or that you're ready to write some other new grant. And it's not at all unusual with academic funding that one result early on tells you, well, that avenue's not going to work at all. I'm going to take the money and just go down this totally different avenue that you weren't funded for, but you're excited about. And if that pans out, you've got a whole new grant you can write when your money runs out. That's where innovation comes from in academics. But in the industry, we've got these milestones, we have to hit them. And if it looks like it's not panning out, the money goes away. And that's a different kind of thing. On the other hand, they have, at least my partner has this project management software that every time we have a conference call, we have a list of action items. And the next month when we have another conference call, we go down that list of action items and see has everybody done what they said they were going to do over the last 4 weeks. And so it's a different type of research. So I wouldn't call it necessarily a challenge, and it's certainly keeping us on track to do exactly what we said we were going to do at the start. But since I've spent my entire career in the academic world, it's different seeing it from the industry side. So I'll stop there.
Yeah, I mean, I would just pile on and agree. I mean, that's the fundamental difference is when you're working with industry, they're very goal-oriented and they expect their reports. If you've agreed to send them a quarterly report, you better send a quarterly report. The other aspect is you have to be honest with your partners. I mean, if you find that something's not going the way it should, there's always the chance that the money would go away. But on the other hand, most companies know if they're funding an academic researcher, unless it's for a very specific assay that they can do or something, they expect that certain things aren't going to work. So if you're honest with them and ideally have a solution or another path to go down, they've already committed the money. If it's reasonable to go ahead and continue, they'll go and they'll go ahead and work through that. So there's a lot more, management and personal interactions and things that you have to do versus just running your lab and telling people what to do and so.
I think communications is one of the big things we've seen as well, is just really making sure that you are communicating with your partners, but like Roger and Keith indicated, honestly and in a timely manner. They do really expect that from us. I want to switch to the other side, and let's talk about some of the benefits that you get besides just the obvious financial support for your projects. Maybe start with you, Yvonne, some of the benefits you get to your lab or collaborators or whatever else that you find you get out of this sort of work.
So much of my career is in industry. I tend to think the other way. So I'm not sure I'm the best person to answer that from an academic lab. What are the benefits? But we have a lot of not just with Andrea, but previously when I was at ICO, a lot of really good collaborations between academics and industry. I think the best is that you have the innovations from the universities and academics, and then you have the very specific task-oriented that can be very efficient. So when a company puts the forces behind, whether it is doing a high-throughput screening or providing cameras or doing all that, you can really accelerate your projects very fast. So I think the best way is because there's no one particular term sheet that is going to be ideal, because each PI and each company have something that they want. So really kind of be honest about how to work out this in a way a marriage. It's like a prenuptial. Like what do we want? What are you going to provide? And it's a negotiation, right? Like any working relationship. And if you kind of work it out and have a good trust, there's fantastic scientists on both ends that really, really is great. Industry has, in the recent years, become like schizophrenic in a way. One year they said they want to work on paint, another year they don't. So it's a little moving target, and so whatever you think they want now may be different. So it's becoming a lot trickier to work with. So, but that said, for better or worse, with no judgment, NIH is moving a lot of focus in terms of the grants and RFA in translational research, whether it is diagnostics, whether it is device, biologics, small molecules. There's specific RFAs that want you to do drug development. I actually haven't personally worked with defense or the Army, although there's RFA I'm going to look at, but they are even more task-oriented, and that may be something that for your work, especially with what you can talk about stroke, but you can talk about TBI and other things that they will be interested in. But it's a totally another mindset that I'm reluctant to actually learn because it's a very different type of mechanism. So I just try all of those, NIH, SBIR, universities, in industries, and depending on what you want to do and what the environment would be at that particular point, you may hit the right target. Like a lot of companies now would only work on neurological disease of genetic, component. So if you happen to work on the particular autistic model that have a genetic basis, then it's a great. But if you happen to work on, something that's not that, then at least at that period of time, until that company change their mind, you'll have to have other, just be flexible, I guess.
Any other benefits that any of you have experienced working with industry?
I don't know about working with industry, but in terms of doing this more translational project, it's been very interesting for my lab group and my students. Especially we get a lot of undergrads who are pre-health career of some sort. And to them, explaining all my basic science stuff, they're like, oh, okay. But then when you can bring out this more applied project, then it makes it, they can make sense of that.
Concrete example?
Yeah, concrete example. So I actually have 4 different undergrads are helping me collect data now with the stroke survivors. 3 volunteers. We're only actually paying one of them, but they're extremely interested in this stuff, and I think it's been a really good addition to my lab.
And you wouldn't have had that with your other research?
No. Also, we've because of the stroke component for the project that was funded by the Johnson Center, I have a collaborator in the occupational therapy department at IUPUI who helped us. We had to train ourselves to do these clinical assessments and all sorts of other stuff that I had never done before. And my students and I got to be exposed to her, which we would not have been if we had not undertaken this project. And that's turned into a useful collaboration for a lot of reasons. I discuss with her my basic science research as well in terms of what could be the occupational therapy application for this. So I would say it's expanded my research quite a bit.
Well, I would just argue that for my specific indication, scientifically it was very useful to be able to have that collaboration and that seed funding from the Johnson Center that allowed you one to say, okay, here's this high throughput screen that was run initially by a postdoc in my lab, And then we submitted a core facility grant that identified some hits for disrupting this protein-protein interaction. Before that time, the only thing that we had available to us to evaluate this target and whether disrupting this target would suppress pain were peptide inhibitors. And peptides are not very good drugs because you can't give them systemically, they get rapidly broken down. So everything that we had to do with these peptides was quite limited. We'd have to inject them locally, like directly into the spinal cord. So that would also prevent us from asking other questions, or just made it more difficult that having a small molecule that could be then given systemically, that could be then given readily with repeated dosing, became very powerful to ask scientific questions that couldn't otherwise be asked. And the industry, of course, had more of the expertise then to, identify and, do the type of things that need to be done with running a very large high throughput screen.
So for my project, it was very advantageous to be working with a large biotech company because they had access to resources that I simply didn't have access to. So one example is I mentioned we were trying to find a way to combat this Asian soybean rust fungus. It's considered such a horrible disease in the U.S. that you have to have what's called BSL-3 laboratory to work with it, which is very high containment. And we don't have such a facility at IU, so I can't even work with this fungus that I'm trying to develop disease resistance for. The company has greenhouses and field sites all over the world, including places where there's abundant Asian soybean rust already. So it's not an issue for them to test out plants that we've put this genetics into to see whether the system works. So they can do the tests for us. They pay for all of that. They've got the facilities to do it. And so that's a huge help to my research. But they also have great resources for what we call genomics, meaning that they have DNA sequence information for this particular pathogen from strains isolated all over the world again. And it's really useful for us to see the diversity that's present within the pathogen, because one of the things that we really focus on is we're trying to develop a way for the plant to recognize any strain of the pathogen. And so we need to find particular proteins that are present in all these different pathogen strains. So again, they have that data. It's not public data. It's data the company has paid for and generated. And so through this collaboration, we have access to that data. So there's been lots of benefits to our research program to be able to work with a company on this project. And it's truly a collaboration. It's not just that they're giving us money to do the work. We're doing it together.
Yeah, I mean, I would like to really emphasize that because I think a lot of faculty who haven't really thought about working with industry before kind of think about it, well, I can go to industry, they can get me money, and that's true. I mean, money helps. You got to have money to run your lab. But I've seen a number of projects where really the most valuable thing they got from the industry sponsor was access to some type of research, because access to databases, access to chemical libraries, maybe access to, a super duper mass spec that nobody else on the earth has. I mean, companies invest a lot of money in very specific areas, and that often can give you a leg up back in your academic life, because you can generate some preliminary data that nobody else can generate because they don't have access to those materials. So it's really, it can often be, those resources can be more valuable than the money they actually put in your lab.
Great. Well, I'm going to begin to wrap us up here, so there's some time for networking and questions and answers. So I'd like to ask the panel to, if you could, give a recommendation of advice or mentorship that you, from your experience, feel would be important to somebody considering entering this sort of work. Well, I'll start with Roger this time. Just picking out of the blue.
it kind of gets back to what I said in the beginning, which is just cultivate your connections. I mean, you think you want to try to reach out to do a translational project with a company. Just think about who you know at that company, or who you know that knows somebody at that company, and meet them at meetings, invite them here for a visit. It's definitely, it really helps to develop a relationship with people at the company so that they are interested in what you're doing and they trust that you have a good idea.
Cultivate your relationship with Keith so you have a
Yeah.
source of unbiased advice.
As I actually have also quite a bit of effort with the Indiana Clinical and Translational Institute, so CTSI, and originally I was a navigator, and now I have this title of entrepreneur in residence, which I actually don't know what it means. But I'm working with the CTSI for the next renewal. So if you have questions about CTSI in general, but a lot of the component of CTSI is actually, we think PI think of it as funding, but actually a significant part of the resources is actually on education. And we think about is creating the workforce, whether it is the undergraduates, the graduate students, the postdocs, or the faculty, that will do quote-unquote translational work, whether it's translational within the university or translational within setting up a company. So we're going to, there are lectures that are going to be live stream about, I think there's one coming up October 31st, 10 to 11, about exactly what it was. I forgot the topic. It was something like industry, how to
Drug discovery.
from academics. I forgot exactly the title, but it will be in MSB2, and it will be live stream, and it's run out from the Indiana. All these acronyms, like basically Indiana Innovation Centers. Anyway, it's an incubator space in Methodist Hospital that is kind of run by CTSI, and that's where Energen and actually Coke and quite a couple of companies are there. So they are kind of like, not exactly, but they kind of like, they are where the IU help people to kind of start company and be entrepreneur. So not exactly, but overlapping goes with the Johnson Center. So all those are resources that you can like utilize to little seed grants, whether it's from the ICBI, something like that, or Johnson, that will give you maybe it's 5,000, 10,000 to kind of just start those ideas, as well as helping in general to connect and find collaborators, whether it's clinicians, the people that like can write the programs or whatever around the 4 campuses. So basically, IU Bloomington, Purdue, Indianapolis, as well as Notre Dame. So if you have a project that you were starting like within the CTSI, we'll be hooking you up with some of the some of the people from Purdue, Notre Dame, which have a huge presence in the agriculture area. So.